Hello children. In our today's lecture, we are going to deal with the topic called as genetic code. When we look into the previous lectures which we have gone through, we were discussing about transcription, that is the formation of messenger RNA from the DNA template strand. Then, according to the central dogma, the next step will be the translation. What is translation? It is formation of a polypeptide chain based on the codon sequence given on the messenger RNA, which has been formed by the process of transcription. So, before we go to understand what is translation, which is the next step after transcription, let us understand actually what is genetic code. If you remember a little bit of information which we have discussed in our form 11, DNA comprises of 4 nucleotide, or you can say comprises of 4 nitrogenous bases, that is adenine, guanine, cytosine and thymine. And these 4 bases are responsible for the formation of genetic codon, meaning that these 4 bases in the form of triplets in the combination of 3 each leads to the formation of genetic code. Let's see one table. In this particular table, you can see that what we have done, that the bases are arranged in 3 positions. You can see the first position where U, C, A, G is there. Another one in the same pattern on the top, second position you have arranged as U, C, A, G, and in the same pattern on the third position U, C, A, G, U, C, A, G, like that it is being arranged on the third position. If you arrange the nucleotide bases in this form, what will happen? In each box you will have set of 3 nitrogenous bases, and these 3 nitrogenous bases will code for one amino acid. For example, first one, if you see, how come it has come? First U comes from the first position, second U comes from the second position, and third U comes from the first U of the box. So triple U, 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 and this is responsible for the formation of a amino acid called as phenylalanine. Similarly, you can see the second one, U coming from the first position, second U coming from the first U, that is from the second position, U1 is there, and the third will be C, which is a second nitrogenous base shown on the third position side of the box. So it will become UUC. Again, this UUC is responsible for the coding of one amino acid, that is phenylalanine. So in this case, what I am talking about is that out of 4 nitrogenous bases, 4 nucleotide nitrogenous bases, only 3 are responsible for coding a particular amino acid. And this particular sequence, if you talk about, I am just reverting this slide, they are, you can say 4 amino acids are 4 nucleotide bases multiplied by 4 combinations multiplied by 4 times. So in total there will be 64 codons present. If you look into this table and if you total up all the triplets which you can see, they will be 64 in number. So there are 64 codons which codes for some 20 amino acids. This is our genetic code. If you talk about the definition, I am just reading this slide. Relationship between the nucleotides on messenger RNA and sequence of amino acids on the polypeptide is called genetic code, meaning that messenger RNA is going to carry these triplets on itself, and once it comes out of the nucleus and goes and attaches itself to the ribosome for the process of translation, and after the translation whichever the polypeptide chain is formed. Now what is polypeptide chain? It is the polymer of amino acids. So amino acids will be linked together in a chain form. So they will be this is going to be called as polypeptide. So this polypeptide, once it is going to be formed, it is called as genetic code. So polypeptide chain formation depends upon the genetic code present on the messenger RNA, which is formed from the template strand of the DNA present inside the nucleus. Second point, there are only 4 bases that we all know: adenine, guanine, cytosine and thymine. They are present in triplet form to make the code with 3 bases in order to synthesize amino acids. That means you have 3 bases and these 3 bases are present in triplet forms, and these triplet together will be synthesizing amino acids. So like this, if you talk about 4 bases present in the whole living system, these 4 bases in different permutation and combination in triplet forms will be converted into 64 codons, and 64 codons will be synthesizing 20 amino acids, and these 20 amino acids in different permutation and combination give huge number of characters in the whole world, millions of characters. So that is how the genetic codes is going to work. Again repeating, the 64 codons are present, that is 4 into 4 into 4, total 64. And this whole theory which I am talking about, this whole table which I am talking about, is given by George Gamow. So this is actually the tabular form which says that the genetic code is in triplet form. The codons are in triplet forms. 3 nitrogenous bases together combines to give 1 codon, and a codon is responsible for the coding or for the synthesizing of amino acids. This is actually the genetic code. Now, what are the features which a genetic code should have or is having, which makes it as a genetic code? Let us understand that thing. So, once we go through the silent features of genetic code, let me make you understand first, and then I go through the slide. So, I am just moving the slide and let us go through the diagrammatic understanding of it. First one. Now we say that there are 64 codons. That means 64 codons are responsible for 20 amino acid coding. But in genetic code, there are only 21 triplets which actually code for amino acids. Remaining 3 triplets, 3 codons, they are called as stop codons, or we call them as terminating codons. These terminating codons, they help in the stopping of the translation. Otherwise, translation will keep on continuing if there is nothing to terminate them. So, in case of this genetic code, there you will be having a start codon and an ending codon. Start codon will be always AUG. Remember this thing very clearly. AUG is called as initiation codon, which is called as start codon, and any formation of this polypeptide chain will start from AUG. Okay, so the first amino acid which will be coded for any polypeptide chain will be from AUG, and the amino acid which is coded by AUG is called as methionine. So you can say that AUG has double functioning. First, like other triplets, it will also code for an amino acid. So the amino acid which is coded by AUG is methionine. Plus it has second function, that it is a initiation codon. So that is one property, that is there is a start codon in case of genetic code. Second one, we have a set of 3 stop codons also, which helps in terminating the translation process. What are they? I am just naming them. One is UAA, second UGA, third UAG. Repeating, UAA, UGA, UAG. These are the 3 codons which are called as stop codons or terminating codons, which will stop the translation process once the translation machinery reaches there. So AUG is the initiation ones, and these three ones I am just repeating again, UAA, UGA and UAG, they are called as stop codon, and it will stop the translation there, making a polypeptide chain complete. This is the first property, the first silent feature of genetic code. I'm telling we go for the next one. Next code says that our amino acid or the genetic code is always unambiguous in nature. Unambiguous meaning what? Only one particular amino acid will be coded from one codon. Meaning, if you can see in the first part of this diagram, GCA is one codon. Now this GCA will code for only one amino acid. It's not that GCA will code for valine also, it will code for lysine also, it will code for tryptophan also. It will code only for one amino acid, meaning one codon is equal to one amino acid. At a time, a single codon will code for one amino acid only, and this property is called as unambiguous or specific property of the genetic code. Second one, it says that some amino acids are coded by more than one codon, and this gives the property called as degenerative in nature. Now for that you can see in the second figure. When you see the second figure, AUU is one codon, AUC is another codon, and AUA, it is the third codon. So all these three codons are coding only one amino acid, that is isoleucine. So what is the meaning of it is that isoleucine is one amino acid which can be coded by different codons, but at a time one codon will code only one amino acid, that is the unambiguous nature, and many codons can code one amino acid, that is called as degenerative nature of the genetic code. So one amino acid can be coded from the many codons, but each codon will be coding only a single type of amino acid. So single type of coding amino acid, that is unambiguous nature, and many codons coding a single amino acid is called as degenerative nature of the genetic code. Third one, we look into the third figure. The genetic code is commaless. Commaless means once you are going to read the whole codons on the nitrogen, on the DNA strand, you will never have a comma or full stop or a hyphen there. It is in continuation. You can see in the third figure, the last point where tick is there, it says AUG, CU, CU, CU. That means three parts will be read. Triplet will be read. So one triplet will be coding one amino acid, another triplet will be coding another amino acid, third triplet will be coding another amino acid. So this will be in continuation. It will be nothing like that, okay fine, AUG is one, then there is a gap, then CUC will be there, then there is a gap, then some other three triplets are there, then there is a gap. No. They are present in continuation fashion and without any punctuation. So that says it is comma-less reading. Third one, this genetic code, when we talk about adenine, guanine, cytosine, thymine in DNA, is universal. All the living system, whether it is a bacteria, whether it is a human being, the most advanced organism on the earth today, both of them, they will have the same type of genetic code. So adenine, guanine, cytosine and thymine will be present even in the bacteria, will be present in the plants, will be present in the animals, will be present in the human beings. So there will be no difference in it. So it is universal genetic code. These are some of the what you can say understanding parts of the genetic code. Now I am reverting back the slide, and the things which I have read or I have made you understand, I am just going through it. First point, silent features of genetic code. The codons are triplet. Out of 64 codons, 61 code for 20 amino acids and 3 codons, as I said, UAA, UGA, UAG, do not code for any amino acids. Hence, function as stop or terminating codon. Second point, one codon codes for only one particular amino acid. Hence, the code is unambiguous and it is specific. Next one, some amino acids are coded by more than one codon. Hence, a code is degenerative or it is degenerate. Next point, the codon is read in messenger RNA in continuous manner without any punctuation. Thus, the code is commaless in continuation, AUG, CGA, and it will keep on moving one after another without any comma or hyphen. Next one, the genetic code is universal, that is a particular codon codes for the same amino acid in all the organism except in few mitochondria and protozoa, that is a kind of an exceptional case. AUG is dual in function, codes for methionine as well as the initiation codon. That's what I said, AUG will be having two purposes. These are the different properties of a genetic code. A little in history about it, I am just moving the slide, the last one. A general information which has to be a little bit remembered if possible. There are names of scientists like chemical method for synthesizing RNA molecule with defined base combination to develop genetic code was given by Hargobind Khurana. This was the person who actually discovered how messenger RNA is formed. Next one, Marshall Nirenberg artificially synthesized protein to understand the nature of codon. So, how the proteins will be synthesized with the help of messenger RNA going to the ribosomes, and then the translation process happens. This whole thing was worked upon by this scientist called as Marshall Nirenberg. Next, history of it, polynucleotide phosphorylase also help in polymerizing RNA with defining sequence in the template independent manner, and this was given by a scientist called as Severo Oka. So, this is actually a little information, this is little history about how the genetic code was discovered and how it was different scientists have given their contribution in it. The main thing to remember is that first thing, the genetic code is triplet in nature, that is a codon is formed of three nucleotide bases. So, like that we have 64 codons present, 61 are functional for making amino acids, 3 are stop codons that will help in terminating the translation process. Third one, our genetic code is unambiguous nature, means one amino acid coding for one codon coding for one amino acid. Second one, our genetic code is degenerative in nature, that means it is many codons can code for one single amino acid, and when the whole genetic code is read, it is without any punctuation. Next point, it is universal in nature, and one codon, that is AUG, functions in dual manner, that is it is initiation codon also, as well as it also codes for a amino acid called as methionine. So, this is the whole crust of the lecture just now which we have discussed. This is all for today. Thank you.